Yeah, so back to my favorite topic, confounding. Yeah, so previously we talked about obviously normalizing for library size, and we have simple strategies that are effective for removing this as a confounder, essentially. Of course, not everything's as well defined as that particularly. And as we mentioned in the sort of identifying confounding chapters prior to normalization, technical confounders, batch effects, and things like this can arise from lots of potential sources. So differences in reagents, isolation methods, the particular lab where things took place, the experimenter who performed experiments, day, time, all this sort of thing can lead to technical confounding between biological signal and other sources of variability that we're not interested in studying per se. And so accounting for technical confounders, having identified them in the first place, and particularly accounting for batch effects, is a very large topic and involves principles of experimental design, and there are certain things that you can never correct for at the analysis end if the experimental design has been flawed in the first place. So when thinking about studies, obviously you hope that someone analyzing data, that you hope that you or someone statistically minded has had input into the study design in the first place to make sure that you're not in a situation where, say, batch is completely confounded with biological group, in which case no statistical trickery can save the day there. But we're going to sort of gloss over all that. There's not time to discuss that in detail today. So instead, we will focus on what we can do on the analysis end in order to take account for confounders, assuming that the experimental design is appropriate in some way. And so fundamentally, what we're wanting to do when we account for technical confounders is identify and ideally remove sources of variation in the expression data that are not related to the biological signal of interest. And various approaches exist, some of which use spike-in or housekeeping genes, some of which don't. And so we'll discuss a few aspects of these in this chapter now. And so between now and the end of the session at 5, this is sort of the topic that we'll cover. So there's time and space to discuss and to think about these things. It can be a sort of, it's an area where there are some highly sort of technical points, which I'll do my best to make clear, but may not be so. And also in the way we do the assessment or deciding on what is effective in terms of some of these batch effect correction methods is a little bit subtle as well. So I'll try to explain them as well as I can, but feel free to chime in any time when things are not clear, because we do have time to discuss these and tease out some of the underlying issues. So that's the plan between now and the end of the day. So we've spoken throughout the day about spike-ins, and the most common version of spike-ins is ERCCs. And so the fundamental idea, of course, is that we apply a known quantity of control genes for each cell, and then we can use that on the assumption that that is consistent across all cells to normalize the expression levels per cell to this consistent sort of set of control genes. And so, in principle, all of the variability that we observe for these spike-in genes should be technical noise, because there's no biological difference, or there should be no difference in input for these control genes. And so, again, in principle at least, we should be able to quantify and possibly remove technical noise by fitting a model to the spike-ins, and in a loose sense, subtracting this technical noise from the expression levels of the endogenous genes. And so there are several methods available that use this basic idea. One of them is a tool called Basics, which fits quite a complicated hierarchical Bayesian model that incorporates information about spike-in or control genes in order to define the technical variability per cell, and then use that in a model that also can be applied to the the expression levels of endogenous genes. There's SCLVM, which we mentioned previously, which focuses more on looking at cell cycle factors, but can also be tweaked to try and identify batches as well. And there's a third tool that we'll mention here called RUV, which stands for Remove Unwanted Variation, which is, of course, the aim of what we're doing. And RUVG was developed for bulk RNA-seq, but can be applied in other settings as well. And the RUV package has several methods that do slightly different things, although based on the same sorts of ideas. And one of them, called RUVG, tries to use control genes to identify technical variability among samples and incorporate that into a model that then identifies and removes the confounding factors. So each of these approaches uses different noise models and different fitting procedures. And so while they're all sort of coming at it in a sense from the same conceptual point of view, there are many differences in the methods that they actually apply to the same problem. So alternatively, one can identify genes which exhibit significant variation beyond technical noise. So you could identify these by sort of distance to the median expression, or there are various approaches to identifying highly variable genes. And these could be used along with the spike-ins. One issue with the spike-ins for normalization, as opposed to QC, which we discussed earlier in the day, and particularly ERCCs, which are derived from bacterial sequences, so are substantially different from human transcripts in some important ways, is that in practice, their variability can, for various reasons, actually be higher than that of endogenous genes. So although in principle, spike-ins and ERCC spike-ins should give us this baseline of control genes that we can normalize to, in practice, that's not always the case. And so I've certainly seen many data sets where the variability of ERCC spike-ins is higher than endogenous genes. And of course, if something goes wrong with the ERCCs, and I've had this happen, that different batches of plates processed at different times, basically some were processed with what we can only assume was a faulty batch of ERCCs, and so our ERCCs gene levels are not at all comparable between batches, which obviously defeats the entire purpose. So they can be useful, but they can't be applied blindly either. So, like with all of the things we've been discussing today, it really pays to take a close look at the behavior of things that you're really relying on as a basis for analysis, and that particularly applies to ERCC spike-ins, especially if you're using something like BASICS or this RUVG method, which put a lot of weight on the assumption that the things you identify as control genes are in fact consistent across your cells, and therefore that the variability you observe is truly technical variability and not something quite different. So given the issues with spike-ins, often we can get better results for identifying batch effects by just using the endogenous genes. There's a lot of information in the endogenous genes, and there are a lot more of them, which is sort of a statistically sounder basis for fitting models, rather than, say, 96 spike-ins or 92 or 48 that might be expressed in a given cell or so on. And there are other methods that don't rely on control genes, but in other sense sort of assess the overall expression matrix of endogenous genes and have various ways, based on this sort of idea of hidden factors or latent variables in a broad sense, for identifying batch effects or other confounding factors that we might be able to identify with a model and then remove. And part of that is because there are a large number of endogenous genes that on average don't vary systematically between cells. And where this sort of variation applies, then these methods can be quite effective. And one of these, which we'll discuss in this chapter, is this RUVS method, so another method from the RUV package. And we'll explore how they work and don't work and compare down below. So a generally sort of challenging question when considering different methods is how to compare them and evaluate them effectively. So when we know about particular batches, we can apply that knowledge to assess how well we're removing batch effects. That's obviously much more difficult if we're trying to learn technical effects that we haven't directly observed. But here with this data set, we know we have samples from 3 individuals that have been each processed in different batches. So we have both inter-individual variation and variation between batches within an individual. And so we'll consider 3 different metrics, which are all, we think, reasonable based on our knowledge of this experimental design. But depending on the biological question that you wish to address, these approaches may need, or likely will need, variation if you're sort of taking your own data set with its own particular characteristics and trying to decide what is an appropriate metric for, if a particular method is doing what you hope it will. Having said that, I think the lessons we can take out of this particular chapter should apply generally, but with the caveat that basically each data set needs to be assessed on its merits, and we can't apply any of these methods blindly and expect perfect results. So this chapter, we're loading up some helper functions. We're loading this RNA-seq package, which has many of these functions, scatter and scran as previously. We're using edgeR and differential expression as one way to assess the performance of different batch correction methods. We'll discuss that later. So we'll load the UMI data again and work from this UMI QC object and identifying endogenous and ERCC genes as previously. So what does RUV do? So factors that contribute to technical noise frequently appear as what we call batch effects, and cells processed on different days by different technicians, as we've discussed, can systematically vary, and this is what we want to avoid. So we're going to consider now this tool RUV-seq, so remove unwanted variation. And briefly, just to give you some idea, it works as follows. So if we have N samples and J genes, then RUV fits a generalized linear model where the RNA-seq read counts are regressed on both known covariates of interest, so that could be things like the total features expressed per cell or other QC metrics, other age, sex, whatever other covariates we have in groups or batches that we know about, and then also unwanted or unknown factors of unwanted variation that essentially the model will try to learn. So in this case, what this equation here is showing is that, so this is, if we didn't have the log here, then we'd sort of be thinking about a normal linear model, which might be more familiar, but here we're saying the log of the expected counts given these parameters is a linear function of some known covariates, which are going to be encoded in this matrix W, so a value for the known covariates for each cell, and some alpha coefficients that we can estimate from the model, some unknown set of factors which we need to learn, both the factors and the coefficients, and offsets that can account for, say, differences between library size and so on, which is what is used to adjust for library size in something like a negative binomial model as used by edgeR or DA-seq2. So the other way around. So W is our unknown factors of variation, and X is the design matrix in the normal sense. So these offsets as well can be like total library size, potentially adjusted by size factors as we've computed previously. And so the challenge here is that simultaneously computing the unknown factors, their coefficients, the coefficients for the known covariates, and K, the correct number of factors, of unknown factors that we want to fit, is not feasible. So instead, sort of up front, we need to say, I want to look for or allow the model to fit K factors. And given that, then we can estimate the factors of unwanted variation, so this W matrix, in various ways. So AUV offers 3 methods to do this. One is AUVG, which uses negative control genes, so that in our setting could be ERC spike-ins that are assumed to have constant expression across samples, so the variation for those genes across samples is only caused by technical variation. AUVS instead uses replicates, so replicate samples, where we say the covariates of interest are constant between these samples, and we'll use that to define sort of our replicate or negative control samples as the basis for batch effects. I'll explain with some examples what that means further down to clarify that. RUVR uses residuals, so it first fits a GLM regression model on the counts on the covariates of interest, and then based on those residuals tries to essentially learn the unknown factors, having removed the effect of the known covariates of interest. So we're going to focus on AUVG since that uses this concept of control genes like spike-ins, and AUVS, which uses the endogenous genes instead, and as we'll see, often performs better. So to actually run AUVG, there's a function called AUVG from the RNA-seq package, and so we can run this. So we supply the raw counts and the ERCC genes, which we've nominated to be the control genes in this setting, and we define K to be 1, and we can then take the normalized counts from the output of RUV and add that to our SCE set matrix in a defined slot. We can run it again with a different K and look at them. Then we can also convert these normalized counts to log counts per million, which, as we've previously discussed, are generally preferable for doing things like PCA and that sort of thing. Using RUVS is slightly more complicated in this case, and so if we look, if we ignore this mess for a second, and instead look at the RUVS command, we can see in these arguments that we need to supply it with the count matrix, as previously, a nominated value for K, and a C index, which is the row names, so essentially the gene names. And here we're using all of the genes, all of the endogenous genes at least, in this fit. And we also need to provide this SCIDX argument, so sort of sample index argument, which defines which samples we're nominating to be replicates. And so that's where this code up here is used to define this. So what we need to define is a matrix that effectively nominates which cell applies to which sample. And so here we're saying that row 1 will contain the indices for this individual. Row 2 will contain the cell indices for this individual, and the third row contains this. I certainly found this a slightly counterintuitive way to apply this information for the function, but that's the way the authors wrote it. So this allows us to essentially nominate all the cells from the first individual as replicates, those from the second individual as replicates, and those for the third individual as replicates. And what we're saying there is that, systematic differences between cells from the same individual are essentially batch effects. They should be systematic, large-scale differences, should reflect technical sources of variability that we want to remove, whereas differences in cells from different individuals, we sort of want to retain because we think that's likely to be driven by biological sources of variability, and we don't want to remove that sort of variability from the data set. Does that make sense? Maybe. Sort of. So to compare it again to RUVG, so in RUVG, we're nominating particular genes, and for those genes, we're telling the model variation across cells in these genes is technical variation, because we've defined a particular set of genes as control genes, as genes that shouldn't change in expression except due to technical sources of variability. In RUVS, instead, we're saying these samples, in our case, particular sets of cells, are in some sense replicates of each other, and variation within a set, one of those replicate sets, should be mostly technical, but we want to retain variation between these sets, because variation between the sets is the biological variability that we're actually interested in studying. And so in this particular setting, we're defining cells from the same individual as replicates, so that hopefully finding variation that differs within an individual, defining that more or less, broadly speaking, as technical, whereas we're retaining variability between cells from different individuals. So we can fit these models, this RUVS approach, with different values of K again, and similarly save the normalized count values into our SCE set object with different values of K, and we'll also define the log counts per million from these normalized counts as well, and save that so we can assess that. So, yes?
To clarify, if I were to run this on, say, something where I identify several cell types, and to look at the differences between those types of cells, I could define the cell types as in the RUVS, basically as batches, and then run that to exaggerate that difference, let's say, or something, or what would that mean?
Yeah, so the question was, if you had, say, a data set where you'd identify different cell types from the data set, could you then say, okay, this cell type, I'm going to treat cells from this cell type as replicates in this sense, and kind of therefore try to remove variation between cells that I've called the same cell type. And this is where it gets tricky, because when we've defined, in identifying the cell types, you're using the information about differences between the cells from the same data set. And if you then try to sort of use them as replicates, it's like double dipping in the data. And so this would be the same sort of problem as if we said, okay, we'll identify cell types and then look for differentially expressed genes between these cell types that we've defined from the data, because by definition, we wouldn't have identified them cell types if there weren't differences in expression between these cells. So I think this approach only works where, from the experimental design, you have a reason to believe that particular cells are, quote unquote, like replicates, or follow the same identical for covariates that you would fit in the model. So in this case, like the individual they came from is like a covariate in the model, and for particular cells, they share the same value of this covariate, whereas that's not true for cell types that you've identified from the expression data itself.
If I wanted to identify a marker for the differences between these cell types, I could use that method to basically exaggerate their difference and use this to try to classify them in future experiments based on this marker.
Yeah. So
probably better ways of achieving that goal.
That's right. Yeah. So it's sort of the markers. So this is it really works for large scale adjustments of the data, whereas when you're looking for marker genes, you want a small number that have like categorically different expression between the cell types. And I think, yeah, as Vlad and Martin suggest, if that's your aim, there are better ways than, say, shoehorning this global approach to that, I think would be the safer approach. Yeah. Okay, so here, so as we said, two approaches to applying this IEV method. So how do we assess if these are doing useful things in this particular setting? So one thing we can do is inspect a PCA plot, which we've come to know and love for this particular data set over the course of the day, and see if we can, do we observe better separation of biological samples with cells from different batches now sort of better interspersed, which would suggest that technical variation has been removed. So if we look at the REVG1, so this is PCA on normalized counts, fitting REVG with K equals 1. Doesn't look spectacularly good here by any stretch. So we've got, PC1 explains 61% of the variance, so we're not getting great separation at all. If we apply the same model but with K equals 2 instead of K equals 1, we see a slightly different picture. So certainly going from K equals 1 to K equals 2, the model is removing some variation. So this component 1 now captures 48% of the variance. So whatever AVG is doing, it is removing some variation when we fit 2 hidden factors as opposed to 1. If we compare that to REVS, then we see that here REVS seems to be doing a better job on the normalized counts. So we here definitely have better separation between samples of better separation of cells from different individuals. It's slightly improved. Yes, there is some separation of the replicates, of course, which is not particularly what we want. So it's doing some things, but perhaps not ideally what we'd like. For instance, if we look at these blue and yellow cells with K equals 1, they're quite distinct. When we go to K equals 2, we start to see more overlap, say, between these two clouds. So we're removing some variation that seems to be bringing replicates closer together a little bit. If we then plot these, or plot the log expression values from REVS with K equals 2, which is, of course, a more appropriate thing to do, then we're seeing nice separation of the individuals. For this particular individual, it looks like we have quite effectively removed that batch effect, since mostly the cells from that individual overlapping each other. But for the other individuals, we're still seeing they're broadly clustering together, but the individual batches per individual are still quite distinct. So of these 4 approaches that we've looked at here, REVG with K equals 1 or 2, and REVS with K equals 1 or 2, REVS with K equals 2 gives better results in this view, although perhaps it's not quite the end point of where we'd like it yet. So as we did with the in the normalization chapter, we can also examine the effectiveness of corrections using this relative log expression that Tallulah introduced, and we can sort of confirm whether or not technical noise has been removed. So if we produce this box plot going from raw counts to REVG K equals 1, K equals 2, and REVS K equals 1, K equals 2, we see that in terms of relative log expression, the distribution for raw counts is very broad. So REVG sort of collapses that middle 50% of the data onto 0, but there's still a lot of outlying cells around that. Whereas with REVS, with both K equals 1 and K equals 2, we've really removed quite a lot of variability from the data and getting, in terms of relative log expression, much more consistency between the cells. But of course here there's not really much to choose between K equals 1 and K equals 1. So K equals 2 with REVS, they really look very similar here. Any questions about either of those, effectiveness evaluation approaches, whether the PCA-based thing or RLA in this case. Not at this point. Okay, so a third way to do it, the final one we'll discuss here, is to look at differentially expressed genes among batches from the same individual. So the idea here is that if we've taken cells from the same individual, and they're nominally the same sort of type of cell, obviously from each individual, then we really shouldn't find differentially expressed genes between batches. Or ideally, if we were to look for differentially expressed genes between batches from a given individual, we wouldn't find any differentially expressed genes. We wouldn't find any significant differentially expressed genes. And so what we're doing here is we're going to take one individual, so NA19101, so from this PCA plot, the triangles, so we're taking cells from this individual, which has the sort of the most overlapping cells from the different batches after the IVSK=2 approach. And so what we'll do is, among the batches of cells, so batches from this individual, we'll see how many differentially expressed genes we can find with different normalization approaches, or adjustment approaches in this case. So here we're going to take just a subset of cells to make things run a bit quicker. Of course, if you were doing this in practice, then you would not want to do that. You would want to retain all your cells to get the most.
So basically, you want to look how the gene is expressed, how the technical variation of the gene expression between these replicas.
So, well, not so much assessing the technical variability, but we're saying under different normalization or adjustment approaches, some will do a better or worse job of removing this batch effect. And so one way of assessing the effect of batch for a given normalization approach, or what's left, is to say, between batches, can we find genes that are differentially expressed? Because if there's no systematic differences between the batches, we shouldn't see any differentially expressed genes, because these are normally, replicate cells or cells under the same conditions. So under this assessment, a better method will have fewer differentially expressed genes between batches when the cells are coming from the same individual. So that's the idea. So, so we've got 3 batches for this individual, and we just sample 20 cells from each of these batches. So we've got now this little subset of 60 cells, 20 from each batch for this given individual. And so we define a design matrix, which just defines the batch that each of these cells came from. And we can just subset that quite easily from that umi.qc object. And so we've got raw counts here. And so we'll use edgeR to calculate DE genes between the different batches, the different plates for this particular individual. And so, as we touched on before, the input data for edgeR and similar methods like DE-seq2 is always raw counts. And so we account for that, as I'll describe below. So what we do, so we fit the linear model, so generalized linear model in the edgeR case. And the test that we do below looks for a difference in expression between replica plate 3 and replica plate 1. So that's just one way of defining differentially expressed genes. If we wanted to, we could do it differently with different contrasts, but I think this is sufficient for this particular comparison. So to use edgeR, that has a data structure called a DGE list, so we construct that as so. So in this case, we supply the counts, so that can just come out of our SCE set object. In this case, we're not applying any normalization factors, so we just set that to 1 for every cell, and we define the group of interest as the batch, as defined above. And so the first step in the edgeR analysis is estimating the dispersion. So this is, in the negative binomial model, the variance for expression is a function of the average expression for the gene and this parameter called the dispersion parameter. And for modeling expression appropriately, the crucial part, it turns out, is estimating this dispersion parameter, which defines the mean variance relationship. And so we supply this estimate disp function from edgeR with this DG list object as we defined it, this design matrix, which we defined above, so that just, it has a one factor, which is the batch. And we include that so that when we estimate the dispersion, we account for potential differences in expression based on batch. So we incorporate that into our estimation. The one main thing when using edgeR on single cell data as opposed to bulk data is that we want to use this argument trend.method and set that equal to none. So at the risk of giving you too much information, but I can't resist, having worked on edgeR for years, what worked in modeling bulk RNA-seq from small numbers of samples was a parametric model where we, and DE-seq does this too, we have an approach to estimating the variance or moderating the variance towards a value that shares information across all genes in the data set. So when you have very small numbers of samples, these empirical Bayesian ideas of sharing information across all of the genes to improve your inference on each individual gene has proven very effective. And so the idea in edgeR is that you could estimate an individual value for the dispersion for each gene, but in small sample sizes that would be very noisy. So instead, say, estimate it for each gene, look at the relationship between those dispersion estimates and average expression, essentially fit a smooth trend defining that relationship, and then squeeze the raw estimates towards this smooth value. And that has the effect of moderating the dispersion estimates, which means that genes that otherwise would have had a very high value for the dispersion get a lower value of dispersion, which increases your ability to find those genes DE, and genes that had a lower than sort of typical for the average expression level value of dispersion get that squeezed upwards, so it's increased, and that has the effect of sort of removing the possibility of false positives. And so, yeah, there's 8 years of literature on this sort of approach now, and it worked very well for bulk RNA-seq. The trade-off is quite different in single cell, where typically now we have hundreds or even thousands of samples or cells, and so we don't need to worry so much about the noisy variance estimates because we now have enough replicates to sort of essentially estimate them more effectively. So what we found in applying these approaches to single cell data is better to turn off this sort of trending and moderation approach and just essentially use the not worry about squeezing towards this smooth trend, just let the model do its thing. So that's what we apply here, and we can look at these dispersion estimates. So the biological coefficient of variation is the square root of the dispersion parameter for the negative binomial model, and if we plot that value against the average log counts per million, then we see this relationship. So you can see some genes have very high values for dispersion, so highly variable. Overall, there's a sort of slight decreasing trend, which again reflects the fact that as we go to higher average expression levels, the biological component is sort of consistent and less influenced by any technical effects. So no, suffice to say, this is a pretty normal looking plot of BCV versus average log CPM for RNA-seq data. So this is not looking too crazy, even though we've applied single cell data to it here. with that vital parameter for the model computed, then we can fit the GLM with the design matrix and what we've estimated here, and then conduct a likelihood ratio test with the function GLM LRT to this fit object, and by default this tests for significance by dropping the final coefficient of the model, which in our particular design matrix has the effect of comparing expression between batch 1 and batch 3 from this individual. And so if we then apply the top tags to this results object, we see the following table. So the genes as ranked for differential expression by p-value or false discovery rate, the likelihood ratio statistic itself, the average or the log counts per million for that particular gene, averaged across cells, and the log fold change, so the coefficient for differential expression between batch 1, cells from batch 1 and cells from batch 3. So even though we're testing for cells between the same individual, we can see here that there are a number of genes that, according to this differential expression test, are quite significantly differentially expressed between cells from different batches. And so sort of really the headline number here is, so by default, so this function says, so what genes are differentially expressed or not at a false discovery rate of 5%. So that's what the default arguments of this function do. And so we can see, according to this, 125 genes are significantly downregulated, 63 are significantly upregulated, and so almost 14,000 have no significant effect. And so if we plot this here, we see these genes in red. So in this plot of log fold change against average expression, these genes in red, sort of on the fringes, are those that are nominally differentially expressed between batch 1 and batch 3 for this individual. So that is the sort of approach we can now apply to different normalization or adjustment approaches as another way of looking at whether, or which approaches are doing better than others in terms of removing that batch effect and that variation that we're not interested in. So if we run the same analysis but with k equals 2 for RUVG, I'll gloss over the details here, but we can explain that later. Need to, again, define the relevant design matrix. So now we include those factors defining unwanted variation from RUV, incorporate that in the design matrix, estimate the dispersion again, get a broadly similar looking plot as previously, go through the same process of GLM fit with the appropriate objects and the GLM LRT, the likelihood ratio test, and again, we can look at the top table of DE genes, and in this case, whereas this was 125 previously and 63 previously, we've seen that with RUVG we have now, at the same significance thresholds, we've reduced the number of DE genes that we're finding between these different batches. And overall this plot looks fairly similar. What we like to see in these sorts of plots is symmetry around that line of y equals 0. If it's not symmetrical, it indicates something's probably gone wrong with the model fitting, but these look okay in that respect. To compare back to this one, we had a bit more sort of odd effects at the very low expression end, whereas these seem to have gone away here. So overall looking reasonably fine from a model fitting sort of point of view. These log fold changes are sensible, and we've discovered slightly fewer DE genes between the batches. So that looks, insofar as that goes, we'd say that RUVG with k equals 2 is better than just using raw counts. If we now try the same thing, but with RUVS, k equals 2, then we can do the dispersion and so on and so on. Same approach again, and when we look at the numbers, now we've got 83 here and 69 here, so sort of similar-ish numbers, I think, to the 94 and 49 here, although slightly more up, slightly fewer down. There, again, this looks more or less the same. But the thing we've done in all of these so far is ignored size factors between the cells, and we would always include these typically in an HR analysis to moderate the library sizes. So we can apply the size factors computed with SCRAN. So we can compute them here, apply them, add them to the DGE list object, run the same thing again, get a similar looking plot of the biological coefficient of variation against average expression, fit the model again, and now we're still finding some DE genes at a 5% FDR, but it's a lower number yet again from what we've seen previously. And if you look at this, we're sort of also seeing slightly tighter sort of funnel, so generally lower log fold change values between the batches, which is sort of what we'd hope to see. So there, sort of, we've looked at these, two major approaches, methods within IEV for trying to remove this unwanted variation due to batch. Looked at the 3 approaches to defining the effectiveness of different approaches, so looking at PCA plots and the separation or not of cells according to batch and individual. Looked at the relative log expression and how that changed across the with the different methods, and then in more detail looking at the number of differentially expressed genes that we find based on these different methods when we're looking for differentially expressed genes within the same individual between batches. So the final thing is an exercise, so to try this similar sort of thing on the read data again. But are there any questions on any aspects of that before we let you do it? Yep.
So particularly in the RUVS, does that perform comparably to what we see including a blocking factor in the HR model, for example? Because you can just specify the batches in the GLM, right?
Yes. So in this case, so the question was, does this IVS factor that we've identified perform similarly to fitting a blocking factor in the GLM? So given that we know the batches in this case, could we just fit that in the GLM? It's likely to perform quite similarly in this case. So yeah, the real utility of finding the hidden factors, of course, is when you don't know about the underlying source of variation. Where you know about batch effects, then yeah, it's sort of explicitly incorporating them as covariates is obviously a very sensible thing to do. Yep. So the question is, if I heard it, was why in this particular example, in this case, why have we set sizes equal 15 for the compute sum factors here? So that defines the sort of the size of the cell pools that the convolution method uses. So we're going to define repeated, cell pools of size 15. That needs to be smaller than, well, we want that to be smaller than the smallest group size in this case, which, since we downsampled the cells to 20 per batch, we want that to be less than 20. So, yeah. But I think from experience of using the sum factors, it's also can be done. You can sort of let the defaults work, and they tend to perform pretty well, the default approaches in my experience. Yep.
You don't have different batches, but you want to look into kind of intra-heterogeneity between the cell types. Can you use a similar idea that you were talking about using HR in order to just compare the differential expression between kind of the cell type that you've defined during your analysis?
So the question is, if we define cell types in whatever way in our experiment, could we then use HR to look for differentially expressed genes between the cells of different cell types? So this is a little problematic, going back to the earlier question, where if we've defined cell types from the expression data, then by definition there must be differences in expression for some genes between those cell types. And so the statistical framework that HR works in assumes a designed experiment. And so any claims for statistical significance are only valid if you have a designed experiment and you're looking for differentially expressed genes between groups that are defined a priori. So the first thing to recognize is that, like, if you do that, the nominal significance levels cannot be trusted. Now, if you're, say, it may still provide you with like a correct ranking according to the negative binomial model of differential expression between them. So in that sense of searching for marker genes, it may give you a sensible list. So if you look from the top of the list down, then you should see things that are, differentially expressed between the cell types. But I know Vlad and Martin have also thought about the identification of marker genes between cell types in more depth, and I think they would agree that if that's your aim, there are better ways to find marker genes than to use, say, an EdgeR approach, which is really designed for designed experiments. Would you agree with that? I think they are nodding, so yes. Yeah.
Can you tell me what the better methods are for finding?
Well, these two could describe the methods that, for example, SC3 uses, and so I think, I'll let Martin discuss that.
Yeah, so it actually depends quite a lot on what your situation is, and we've sort of developed sort of two different types of methods. So one, which is part of the clustering method that Vlad developed, which is it sort of looks for the genes that are sort of the most unique to each group of cells, sort of after having identified the groups of cells. And it's sort of based around quite a straightforward idea of a sort of trying to find the binary classifiers using only that gene to separate this one cell group of interest from everything else. So it ranks the genes based on how well they perform this classification task. And then we have the opposite approach, or not the opposite, but the complementary approach, which is sort of Tallulah's method, which is assumes that you don't know what the groups are a priori, and that method, which we'll also be talking about tomorrow, identifies the genes that are most likely, or seems to be the most informative for splitting into groups, splitting the cells into groups, and so which method to use sort of really depends on sort of what your framework is and what your question is and what you want to be assuming. And then there are sort of, in this sort of more traditional framework that we've been used to working in with bulk RNA-seq, where sort of, for example, EdgeR and the DE-seq operates, where you assume that you have your known groups and then find the most differentially expressed genes. There have been several methods developed by other groups that we'll also be discussing tomorrow, and you can try to find out for yourself sort of how well you think they perform. Yeah, so I think this is pretty much the last chapter for today. Well, it's a big exercise, right? Yeah. So it will be the usual thing where you work your ways through the exercise of using the reads instead of the UMIs for dealing with the confounders. So and then you can basically sort of once you're done with that, you can head out of here, and we'll see you at 9:30 tomorrow.
Yes. Why did we apply it in the final step, but not previously? Oh, just. Yes, and in general, it makes sense when fitting any EdgeR models to include size factors if they've been computed. So I'll leave it as an exercise to you to explore the differences. Yeah, no, if I were actually to apply this to this sort of problem, I would use the final one of actually making use of the size factors in fitting the edge model. Yeah. I think so, yeah. I mean, I don't know exactly how the syntax would work, but yeah, absolutely, because you might need to define the size factor slightly differently, but in principle, it operates in the same way, and the size factors are used in the negative binomial model in the same way, in the sense of scaling the library size, which comes into the GLM as an offset. So that will work the same way. DSEQ, yeah, I think. Yeah, I don't know in the latest sort of versions if it does more internally by default or not. Yeah, I'm not an expert on that package, but yeah. Okay. I'll give you a bit of time to work through that, and then we'll come back in 10 minutes or so and discuss.